going to take a look at the process of DNA replication. There are a few things to keep in mind with DNA replication. One of them is that it is happening during S phase of the cell cycle. It is semi-conservative, meaning that half of the strands are going to result or be in the daughter strands. The other half of those daughter strands is brand new DNA. The replication forks are what you call it when the DNA splits. You can actually see a nice replication fork right here as well. The replication forks travel away from one another, away from an origin, and as they're doing that, they make a sort of replication bubble. And then the other thing to keep in mind is that when you add a new nucleotide to a pre-existing nucleotide, you can only attach it to the 3' end. You cannot add a nucleotide to the 5' end. It just doesn't work that way. So now the enzymes of DNA replication. There's a really nice animation called DNA replication fork. If you search for that and possibly also McGraw-Hill, that will take you right to that animation. So we're going to look at helicase, DNA polymerase 3, RNA primase, RNA primers, Okazaki fragments, DNA polymerase 1, DNA ligase, and some single-stranded binding proteins if we're lucky. So the first thing that needs to happen is helicase is basically going to come in here and break the hydrogen bonds between the two existing strands. Now, the single-strand binding proteins will attach and keep these two strands from re-annealing, or that's a fancy way of saying coming back together again. At this point, we can separate these two daughter strands into the leading and the lagging strand. The leading strand, conveniently, can be built from 5 towards 3, which is how the direction you have to build DNA, and that happens to be towards helicase. The other strand has the 3' direction heading away from helicase, which makes the lagging strand a little bit more complicated. For the leading strand, really all you need to worry about is the fact that starting at what was originally laid down as an RNA primer, not pictured here, DNA polymerase 3 simply attaches to the DNA strand and builds a complementary strand out of free nucleotides following helicase, la-di-da, and it's done. There we go, done. The lagging strand takes a little bit more machinery to make this happen. First of all, RNA primase is going to follow helicase and leave these little RNA primers sort of along the way in these intermittent points. These are going to be starting spots for DNA polymerase 3, which we actually saw up here as well. It's the same enzyme on the other strand. And it needs these RNA primers so that there is a pre-existing nucleotide to add to. You can't just add a nucleotide to nothing. You have to add it to a 3' end of something. So DNA polymerase 3, floating around, random molecular motion, is going to attach the RNA primer on the 3' end and make its way down towards 3' to the next RNA primer. Once it gets there, it will leapfrog backwards. I should have shown this one going this way. Here, it will make it this way. There will be a DNA strand behind it, and then it leapfrogs backwards in order to follow helicase. So it's sort of taking a few steps backwards, but then leapfrogging forwards, and that way, overall, it does follow helicase. So once it does that and adds more strands, we need to call these Okazaki fragments because they were discovered by a man named Okazaki. They're separated by RNA primers, which is no good. We need to get rid of those. DNA polymerase 1 comes in, attaches to the strand, and simultaneously removes the primers and adds DNA in its place. So you notice there's no RNA primer here anymore. The last thing to take care of is getting DNA ligase in here so that we can attach these Okazaki fragments that are previously not attached to one another and connect the last couple nucleotides from 5' to 3' carbons on the nucleotide. And we are done. So here is a slide that sort of summarizes all the jobs of those different objects throughout DNA replication.